Hello everyone, welcome back to the session. So in today's class we are going to start the mitochondria in cell biology. Okay, so we are discussing about the CUET PG exam in the zoology subject. So in that particular subject we have started an important chapter called the cell biology. So whenever we are discussing about the cell biology, we have completed what will be the basic ideology about the cell and what will be the cell theory and the difference between the prokaryotic and eukaryotic cell organization, and also we have started the plasma membrane and we have completed thoroughly in the line of the exam, and also we have completed the nucleus, so in nucleus as well as the chromosomes. Okay, so all the important factual information as well as the conceptual information regarding with this exam we have completed so far. Okay, so in today's class we are going to discuss about the mitochondria cell organelle. Okay, so whenever I am saying the name mitochondria, you would have always remember the powerhouse of the cell. Okay, but there is so much of interesting as well as an important information regarding this mitochondria is there. Okay, we are going to discuss about that, right? Okay, fine. So the first thing, so you should know what will be the historical background of this mitochondria. Okay, what will be this, who are all the scientists have been identified and give information about the mitochondria. So in that case, the first scientist will be the Colliker. Okay, in 1880. So in 1880, Colliker is the scientist. He observed the mitochondria in muscle cells. We can expect the question like this. Okay, so the first mitochondria will be observed first in which cells, in which type of cells. Okay, so that will be the muscle cells. Okay, and that will be done by the Colliker in 1880, right? Next, and one more important thing will be the other name of mitochondria. Okay, now we are calling this particular organelle as mitochondria, but before the name mitochondria there is another name which is called as a bioplast. So we can also expect the question from here. Okay, so bioplast will be the name of the organelle mitochondria again. So if the question asked bioplast is considered as the bioplast, we can say the Krebs cycle will be taking place in the bioplast. Okay, so you should not think so Krebs cycle will be taking place in mitochondria, but here this is the wrong statement. You should not think like that. You should know that bioplast is also known as the mitochondria. And who named this particular organelle as bioplast is Richard Altman. Okay, so Richard Altman is the name of the scientist who have named the mitochondria as bioplast before the name mitochondria. Okay, now after that, who coined the term mitochondria by replacing the word bioplast? Okay, so that is Carl Benda. Carl Benda is the name of the scientist. Carl Benda is the name of the scientist coined the term mitochondria. Okay, so this is very important thing. Okay, so that we can expect the question from here, the historical background. Now you should know what will be the general characteristics and what will be the size and what will be their shape of mitochondria. Okay, so first you should know the general characteristic features of mitochondria, that it is present in all aerobic eukaryotic cells. See, for the first class I have explained you about the cell theory. Whenever we are studying about the cell theory, we have studied about the endosymbiotic theory. Okay, see here, initially prokaryote is the first organisms have been originated, and after that from the prokaryotes eukaryotes have been originated. Okay, so in the eukaryotes, so the eukaryote, initially the first formed eukaryotes do not perform the photosynthesis and they are not in an aerobic condition. Okay, so for that this eukaryote is having interacting, it is made a symbiotic relationship with some other organisms. Okay, so that is what the eukaryotes is having, the symbiotic relationship between the aerobic bacteria and the photosynthetic cyanobacteria. Okay, so this this is what an eukaryotic organism. So inside that we can see the cyanobacteria and we can see the aerobic bacteria. So with the presence of the cyanobacteria, that eukaryotes can perform the photosynthesis. In the presence of aerobic bacteria, they can survive with the oxygen. Okay, so now, so this cyanobacteria is the one evolved into the chloroplast, and the cyanobacteria is the one responsible for doing the photosynthesis, and the aerobic bacteria, which means the cyanobacteria, will be evolved into the chloroplast, and the aerobic bacteria will be evolved into the mitochondria. As the time goes on, this mitochondria and chloroplast will be fixed within that particular organisms. Okay, so that is why we are saying this mitochondria is present in all aerobic eukaryotic cells. You should not think why it is aerobic, why not anaerobic. Okay, so that is what I just relate this mitochondria with the endosymbiotic theory, that where the aerobic bacteria is the one evolved into the mitochondria. So that is why it is present in all aerobic eukaryotic cells. Okay, and you should know that, see here, okay, see here, this mitochondria is absent in RBCs. Okay, see, also I have explained you that the cell doesn't have the cell, all that we are having uninucleate cell which has one nucleus. Okay, and also we are having multi-nucleus in a cell. Okay, in a cell we are having many nucleus within it, and we are also having non-nucleus. Okay, which means what? RBC is a cell which is not having any nucleus within it. Okay, and also this RBC doesn't have mitochondria. So you should not think mitochondria is considered as a powerhouse of the cell. So you should not think this mitochondria is present in all the cells in our parts in the body. Okay, so in that case that statement is wrong. Why? Because this mitochondria doesn't need in case of RBC. Okay, the RBC doesn't need the nucleus itself. Okay, why? They need more space because the hemoglobin has to carry the oxygen and it has to deliver to the all parts of the body. So that is why it needs more space. Okay, and most of the important organelles as well as the nucleus doesn't present in case of RBC. Okay, see, in mature RBC the mitochondria is absent. This is very important, and the mitochondria is a rod shape. It is in oval and it is spherical shape. Okay, so as it is present in rod shape, oval shape and spherical shape, we can call this particular shape as a pleomorphic shape, which exhibit in variable shape and size. Okay, now its number depends on the energy requirement of the cell. Okay, so energy requirement of the cell. See, for an example, so we are having many cells: liver cell, lung cell, kidney cell. Okay, so in that case the muscle cell always need more energy. Okay, and we know this mitochondria is a powerhouse of the cell. As this mitochondria is a powerhouse of the cell, they are the donor of energy, and the muscle cells are always in need of an energy because we are moving the hand here and there. Okay, and the muscle needs to contract and relax. For all these processes they are in a constant supply of water, they are in constant supply of ATP. Okay, and that will be provided by this mitochondria. So that is why the muscle cell have more number of mitochondria. Okay, now what will be the muscle cells having the many mitochondria when compared to all other cells. Okay, so see here, this will be the structural organizations of mitochondria. So what will be the structure of the mitochondria? So you should know that is a cell organelle which is present inside the cell. This is a basic understanding. And the second step will be, as this is an organelle, you should know whether this will be the membrane bound organelle or non-membrane bound organelle. If it is a membrane bound, whether it is a single membrane bound organelle or double membrane bound organelle. These are all an important thing. Okay, so see here, this mitochondria are double membrane bound organelles. Okay, see, this will be the structure. We are having an inner membrane and we are having outer membrane. Okay, as this is having a two membrane, we should know this mitochondria will be the double membrane bound organelles. Now the first, as we have studied, this mitochondria will be the double membrane bound organelle. You should know what is the importance of this outer mitochondrial membrane. Okay, so this outer mitochondrial membrane, so it is very smooth. Okay, so it is very smooth and it is highly permeable. It contains porine proteins, allows diffusion of molecules which is less than 1000 Daltons. Okay, see here, this is an outer membrane. Okay, and below the outer membrane we can see the inner membrane. Okay, and you should know what will be the speciality of this outer membrane. So we have studied about the plasma membrane, and when it comes to the permeability, this plasma membrane is selectively permeable. Okay, so this is the second thing you have to keep in mind. So first thing will be whether it is a membrane bound or non-membrane bound. If it is a membrane bound, you should know whether it is a single membrane or a double membrane. And the second thing is, if it is a double membrane or single membrane, whether it is selectively permeable or highly permeable or impermeable. This is the second thing you have to keep in mind. See here, it is very smooth. Okay, first thing will be it is very smooth, and the second important thing will be it is highly permeable. If it is a highly permeable, you should know there is a lot of pores within it, and that pores is made up of a porine proteins. Okay, and these are all the pores, and we have studied it is highly permeable in nature, and you should know. So if it is highly permeable, what are all the molecules present in the cytoplasm? Everything will be taken by the mitochondria. Okay, but there is a checkpoint. Okay, what is the checkpoint here? Those molecules which are less than 1000 Dalton, that can be easily enter into the outer mitochondrial membrane. So this is an important thing. See here. Now, what will be the function of this outer mitochondrial membrane? Just now we have studied it is smooth and it is highly permeable, and it can enter and it has a porine protein. So the main important role performed by this outer mitochondrial membrane will be there is a free exchange of ions and metabolites, right? So this is an important thing. See here, so this is an outer mitochondrial membrane. Now the second thing, the inner mitochondrial membrane. See, the inner mitochondrial membrane just opposite to the outer mitochondrial membrane. See here, it is highly selective and it is impermeable in nature. Okay, so what we have studied with respect to the outer mitochondrial membrane, it is very smooth and it is highly permeable, but this is very opposite. It is highly selective and impermeable. Okay, so which means what? It is selectively permeable in nature, right? So if it is impermeable, for what are all the molecules it is impermeable for? See here, it is impermeable to most of the ions and small polar molecules. So these molecules cannot be enter into the inner mitochondrial membrane. And another important thing, this is where we can expect a question. Okay, so this inner mitochondrial membrane is made up of an important lipid layer that is called as a cardiolipin. Okay, so this is where we can expect a question. Okay, so it maintains a proton gradient. Okay, so proton gradient. So we have studied that as it is an powerhouse of the cell, how it can produce a powerhouse, how it can produce an energy. Okay, ATP, just by because of this electrochemical gradient. Okay, so that is what this proton gradient. All these will be discussed detail whenever we are discussing about the cycles. Okay, now what will be the role? So it is a site of this oxidative phosphorylation. Oxidative phosphorylation is the synthesis of energy. Okay, so for that it needs an oxidative state. Okay, oxidation reaction will be taking place in the inner mitochondrial membrane. So this inner mitochondrial membrane is the site of the oxidative phosphorylation. So if the oxidation phosphorylation has to be taken place, definitely there will be the shifting of electrons. As there is a shifting of electrons from one electron carrier to another electron carrier, there will be a loss of energy. Okay, so this loss of electrons, loss of energy will be, this energy will be converted into an ATP. Okay, and that ATP is an energy form. Okay, so this is what an important thing with respect to the inner mitochondrial membrane. Right, now the third important structure will be the cristae. Okay, so cristae means we have studied about the inner mitochondrial membrane. So see, we are having an inner membrane and we are having an outer membrane. Between these two, inner and outer mitochondrial membrane, we are having a space between the inner mitochondria and outer mitochondria. Okay, so that space is called as an intermembrane space. Okay, so there is a folding. This is the folding of the inner membrane. Okay, there is an inner membrane and the inner membrane is undergoing a folding, and that folding structure is called as a cristae. Okay, why the inner membrane has to fold? Okay, to increase the surface area. Okay, so the foldings of the inner membrane, we are calling that particular structure as a cristae. And why it has to fold the inner membrane? To increase the surface area. So that is the main reason why the inner membrane undergoing a foldings, right? Now it contains, what are all the things it contains? So it contains an electron transport system enzyme. So this is an important thing you have to keep in mind. Okay, so in the inner membrane, inner mitochondrial membrane, what do we have studied? It is a site for oxidative phosphorylation. And the cristae here, what are all the things they are having? What are all the enzymes required for oxidative phosphorylation and electron transport system? All these enzymes will be accumulated in the cristae, then in foldings. Okay, and also another important thing where the in which the cristae has is ATP synthase complexes. ATP synthase complex is nothing but F0 F1 complex. This F0 F1 ATPase. Okay, so the F0 F1 ATPase is the one, it is also called as the ATP synthase complexes. This complex is involved in the synthesis of ATP. Right, now what will be the important fact with respect to this cristae? So they are having more cristae. As they are having more cristae, they are having higher ATP production. Okay, more cristae, it leads to higher ATP production. Why? Because this cristae, they are having an electron transport enzymes and they are having an ATP synthase complexes. Okay, see, this is how, see, this portion is called, see, all these brown color things which is present in the cristae, so that we are having an F0 portion and they are having an F1 portion, which we are calling them as the F0 F1 ATPases, right? It is also called as a ATP synthase complex, right? So this is very, very important thing. Now next, intermembrane space. So in this intermembrane space, it is a space between the outer membrane and the inner membrane. Okay, so there we are having an outer membrane and we are having an intermembrane. Between these two membrane there is a space, and that space is called as a intermembrane space. Okay, now see here what this intermembrane space contains. Okay, see here they are having a protons, that is H plus ions, during a respiration. During a respiration process, all the protons will be accumulated, will be stored in the intermembrane space. So this H plus is the one which is creating a proton gradient in the inner membrane. Okay, at the time of respiration process, right? Now the mitochondrial matrix. So this mitochondrial matrix contains a proteinaceous fluid. Okay, so it is containing the proteinaceous fluid, and they are having an enzymes of Krebs cycle. See, this matrix is an important structure when compared to all the structure in the mitochondria, because in the matrix they have contains a proteinaceous fluid. Okay, so in the matrix region, which is a matrix region, so this yellow color, this is called as a matrix region. Okay, so in this matrix region, what are all the things they are having? They are having a proteinaceous fluid. Okay, they are having a proteinaceous fluid, and they are having an enzymes of the Krebs cycle. Okay, they are having an enzymes of the Krebs cycle, and they are having circular DNA. They are having circular DNA. See, so very, very important thing when it comes to the mitochondria will be, so usually the genetic material DNA will be present in the nucleus. No doubt in that. Okay. Now, so in the nucleus we are having a genetic material. Besides the nucleus, there are other organelles which are also having the DNA within it. One is plastids. Okay, chloroplast. Okay, plastids and the mitochondria. Okay, so in the chloroplast and the mitochondria, they are having their own DNA. Okay, now what is the difference between the nuclear DNA and the mitochondrial DNA? Nuclear DNA is linear in structure, and in case of mitochondria, it is circular in nature. Okay, so it is linear, whether where the nucleus having the DNA is linear, and the mitochondria, they are also having a DNA which is circular in nature. Okay, this is very important, and we are having an outer membrane, inner membrane, cristae, matrix, all these regions were the structure of the mitochondria. Where exactly the DNA is present is this mitochondrial matrix. Very, very important thing. Okay, and the second thing will be, as this nucleus is having their mitochondria, we can call that particular mitochondria as the self-replicating organelle, or we can say the autonomous organelle. Okay, semi-autonomous. It is not a completely autonomous organelle. It is semi-autonomous organelle. What is meant by autonomous? It can functions of its own. Okay, they have the capacity to synthesize their protein of their own without the involvement of nuclear DNA. This is what an autonomous with respect to mitochondria. Okay. So what is semi-autonomous? See, even though mitochondria, they are having a DNA of its own, it can has the capacity to produce only up to certain extent, with the where we can say that 16% of the protein. See, what will be the ultimate goal of DNA? So DNA has to be replicated, that is DNA replication, and it has to synthesize an RNA. That process is called transcription, and the RNA has to be converted into protein, that is translation. So the ultimate goal of DNA is to synthesize protein. Okay, now, so this mitochondria, they are having a circular DNA, and now they are not completely producing all the required protein for the mitochondria. Anyhow, they are also need a protein. They are also the organelle which depends on the nucleus for their protein synthesis. Okay, so that is why we are saying mitochondria as the semi-autonomous organelle, not a complete autonomous organelle. Okay, so this is an important thing. And another important thing will be the ribosomes. What will be the ribosome? So in the first class itself, in the second class, we have studied about the 70S ribosome and 80S ribosomes. Okay, in the mitochondria, what ribosome we can say? 70S ribosome will be seen in case of mitochondria, not 80S, right? Now, and it is also having the RNA, it is also having the tRNA, because they are also involving in the replication process, transcription process, translation process. Okay, so then only the mitochondria can synthesize their protein, as it is having the DNA. Okay, now they are also having the metabolic intermediates. Okay, all the metabolism process, they need some substance. Okay, so that all the metabolic intermediates will also be present in case of this mitochondrial matrix, right? So what we can expect in the question? See here, so what do we have studied? Krebs cycle. We have studied the Krebs cycle will be taking place in the mitochondrial matrix, which means all the enzymes required for the Krebs cycle is present in the mitochondrial matrix. Okay, but there is an exemption. Okay, succinate dehydrogenase is an enzyme required for the Krebs cycle. One of the process we need for one conversion we need the succinate dehydrogenase complex. Okay, but this enzyme, what we have studied, all the enzymes required for the Krebs cycle will be present in the mitochondrial matrix, but the succinate dehydrogenase is also the enzyme involving in the Krebs cycle, but this enzyme will be present in the inner membrane. Okay, so this is an important thing, right? Next. Now, elementary particles. Elementary particles, see here, this elementary particles is also called as the oxysomes. What is meant by this oxysome? So, this oxysome is also called as this F1 particles. F0, F1 complex. We have studied that F0, F1 complex. Okay, right? See, this F1 particles and F1 particles, F0, F1 complex is also called as an ATP synthase. So, it is also called as this oxysome. Don't confuse with it. See, what do we have studied? The mitochondria is also called as the bioplast. Okay. Now, this F0, F1 complex is also called as an ATP synthase complex, and it is also called as an oxysomes. Okay. Don't confuse with it. So, this elementary particles, that is an oxysome, we are also called them as the F1 particles. Why F1? Because it was discovered by this Fernandez Morin. Okay, so that is why we are calling them as a F1 particles, right? So it is also called as an oxysome. What will be their structure for this oxysome? The base portion, this portion is called as an F0 portion. Okay, so this will be embedded in the inner membrane. right? This base F naught will be embedded in the inner membrane, and there is a stalk region and there is a head region. So the head region is called as an F1 portion, and the base region is called as an F naught portion. Okay, so this is what the structure of the oxysomes, right? Now, what will be the function of this oxysome? ATP synthase present in the head region. So this is what an enzyme which is involving in the ATP synthesis. Okay, so what we have studied? What will be the site of the oxidative phosphorylation? We have studied. The site of oxidative phosphorylation will be inner mitochondrial membrane. Okay, so the oxidative phosphorylation takes place in the membrane region. Now, so this ATP synthase present in the head region. So this is the one responsible for the oxidative phosphorylation, so that we can get the ATP. Okay. Now, what will be the component? What will be the chemical composition present in the mitochondria? So, what are all the components? We are having a proteins. Okay, mitochondria is having the protein, lipid, RNA, DNA, and also the enzyme. See, the most of the component, so we are having these much of chemical composition. When compared to all these things, protein component having the higher percentage. Okay, protein component having the higher percentage, that is approximately 73%, and the lipid component, they are having 23 to 30%, that is cardiolipid. Okay, and this RNA will be 5 to 7%, and DNA will be the traces means small amount, and the enzymes will be present in the mitochondria will be approximately 60 types. Okay, so this is the thing. Okay, so protein is higher when compared to all other chemical composition. Protein is higher, that is approximately 73%, and enzymes will be approximately 60%. 60 types of enzymes we can see in the mitochondria, right? Fine. Now, what will be the functions of this mitochondria? What will be the function? Of course, it is ATP synthesis. Okay, ATP synthesis, cellular respiration and fatty or acid oxidation. So this is an important thing. See, kindly make a note of it. This fatty acid oxidation is done by the mitochondria. Okay, and you should know this fatty acid oxidation is not only done by this organelle called mitochondria, but the fatty acid oxidation is also done in another organelle that will be dealt in the future classes. Okay, so just for now, just know that fatty acid oxidation will be done in the mitochondria, and this is very important. Okay, now heat production. Wherever the metabolism is taking place, whatever be the chemical reaction taking place, always there will be the heat will be lost as a result of a reactions. Okay, so now this heat production will be brown fat. So just now what we have studied, fatty acid oxidation. Whenever we are oxidizing the fatty acid, the heat will be produced. Okay, now apoptosis regulation, the very important function of mitochondria will be this apoptosis regulation. Apoptosis in the sense cell death, programmed cell death. That is what an apoptosis is. Whenever the apoptosis regulation, the cytochrome C is an important component which is involving in the cell death. Okay, so all these were the functions of mitochondria. So we are calling this particular mitochondria are called as a powerhouse of the cell. Okay, so this is very important thing. Now, mitochondria with respect to the genetic systems. Okay, genetic systems, they are containing a circular DNA. As we have studied before, this mitochondria is having the circular DNA, and also they are having 70S ribosomes, just similar to the prokaryotes. Okay, supports the endosymbiotic theory. Just now we have recalled the endosymbiotic theory. In this endosymbiotic theory, what do we have studied? The eukaryotic cells is made symbiotic association with aerobic bacteria and the photosynthetic cyanobacteria. This aerobacteria is the one, as the time goes on, it evolved into the mitochondria, and now the mitochondria will be fixed in all the upcoming organisms. So this is what the endosymbiotic theory. Now, and the replication and biogenesis process. So the replication and biogenesis means, in the sense, see here, as the DNA is present here, so this is multiply, which means the mitochondria will be multiplied by binary fission. Okay, so this mitochondria will be multiplied by binary fission, so this can be expected in the question. Now, empty mitochondrial DNA replication. Wherever the DNA will be present, and the DNA has the capacity to replicate, transcribe and translate. Okay, so that is why this mitochondrial DNA is undergoing a replication process and strand displacement model. And the name of the model here will be the strand displacement model. And see here, this mitochondria are semi-autonomous organelle, as I have mentioned you before. So this is a semi-autonomous organelle. Why? Because they are having a DNA, and this small amount of DNA has the capacity to synthesize a protein. Okay, and that protein, so even though they are having a DNA, even though they are have the capacity to synthesize a protein, they can't live without the help of the nuclear DNA. Okay, so that is why we are saying this particular organelle as a semi-autonomous organelle. Now, what will be the inheritance pattern of the mitochondria? See, very important. So usually the inheritance will be from both the mother as well as the father. Okay, so this is what an inheritance pattern. So this organelle is inherited only from the mother, not from the father. So maternal inheritance, we can call this particular term as the maternal inheritance. This is the paternal mitochondria are destroyed after fertilization. So this is what an inheritance pattern of this mitochondria. Very, very important. Now the evolutionary importance of this mitochondrial DNA. How the mitochondrial DNA is getting evolved. See here, this mitochondrial DNA mutates. It mutates and 5 to 10 times faster than the nuclear DNA. Okay, see here, the 5 to 10 times faster than the nuclear DNA. So you should know that the mutation rate with respect to the mitochondrial DNA and nuclear DNA, mitochondrial DNA are 5 to 10 times faster than the nuclear DNA with respect to the mutation rate. Okay, so this is very important. So that is why, as it is mutate faster, it is very important to trace the human evolution. And another important keyword will be recent. To study recent evolutionary timelines, mitochondrial DNA will be more suitable when compared to the nuclear DNA. Kindly make a note of it. You should not get confused with the nuclear DNA and mitochondrial DNA. We know that mitochondrial DNA mutate faster when compared to the nuclear DNA. As this is mutates faster, to study the recent evolution, mitochondrial DNA is very suitable when compared to the nuclear DNA, right? Very important. Now, this is used in population genetics and phylogenetics analysis. Okay. Now, so in the exam, how can the question will trap us? Okay, see here, Krebs cycle location. Krebs cycle location will be in the mitochondrial matrix, no doubt in that. Just now we have studied. And electron transport system location in the inner mitochondrial membrane. ATP synthesis will be taking place in the cristae. And why ATP synthesis will be taking place in the cristae? That is why, because of the presence of an oxisome. And oxisome means F0 F1 complex. As it is present in cristae, ATP synthesis will be taking place in the cristae. Now, what will be the shape of the DNA present in the mitochondria? That will be circular rather than linear. Okay. Now, type of ribosome present in the mitochondria will be the 70S. And what will be the inheritance pattern of the mitochondria? That will be maternal in nature, and it is semi-autonomous. Okay, it is not autonomous. Instead, it is semi-autonomous. Okay. See, another important thing, cardiolipin. Cardiolipin is present only in the inner membrane, not in the outer membrane. Just make a note of it. This is very important. And see here the succinate dehydrogenase complex. So what we have studied before, the enzymes required for Krebs cycle will be present in the inner mitochondria, which means in the matrix region. There is an exemption of the succinate dehydrogenase. Why? Because the succinate dehydrogenase is membrane bound enzyme. This is have the capacity to survive in the membrane region, so that is why it is present in the inner mitochondrial membrane. Okay, porins is the one bacterial origin evidence. So we have studied about the endosymbiotic theory. Endosymbiotic theory suggests that aerobic bacteria is the one evolved into the mitochondria, and the evidence for that evolution is this porins. They are saying like that. Okay. Now see here, mitochondria are double membrane bound organelle. We know that generate ATP. How they are generating ATP? By oxidative phosphorylation. Okay, so this is an important keyword. Okay, now that's all about. So far we have completed what are all the important things which can be expected in the examination. Okay, so now we can start the question. So which are the following statements regarding the outer mitochondrial membrane are correct? Regarding the outer mitochondrial membrane, okay, and they are asking the correct statement. Always have the habit of reading the question twice. Okay, so they are asking the correct statement with respect to the outer mitochondrial membrane. It contains a porine proteins. Okay, it contains porine proteins. It is impermeable to ions. It allows diffusion of molecules less than 1000 Daltons. It is rich in cardiolipin. Okay, see, it is rich in cardiolipin. Just now we have studied cardiolipin will be present only in the inner mitochondrial membrane, not in the outer. So we can identify easily the option 4 is wrong. Okay, so you should know, if you find any one option is wrong, try to eliminate the options. 4 is present in B, C and D. So this is what an option elimination technique. This can save your time, as well as you can easily come to the question without reading the other options too. Okay, so the right answer will be option A. Now we are going to study all the statements, but in exam, to save the time, you can use this technique, right? Now, it allows diffusion of molecules less than 1000 Daltons. Okay, so this is a right statement, and it is impermeable to ions. Impermeable to ions is which layer? Inner mitochondrial membrane. So we can also eliminate this second option. It contains the porine protein. As it is containing the porine proteins, it is highly permeable in nature. So the answer for this question will be option A. Now, the second question, this is the mass of following question. So the cristae. Cristae can be matched for matrix and F1 particle and outer membrane. Okay. See, ATP synthase will be present in where? In the ATP synthase, Krebs cycle enzymes. Krebs cycle enzymes will be present in the matrix region. Porins will be present in the outer membrane. F1 particle will be the electron transport system, and cristae will be the ATP synthase. So the correct answer will be option A. 1 minute. Sorry. See, the cristae, they are involving in the electron transport systems. Matrix, no doubt Krebs cycle enzymes will be present here. F1 particles, ATP synthase, outer membranous porins. Okay, so this is very important thing, match. So whenever you find the match of following question in your exam, don't hesitate to attend the question. Try to attend the question, okay, because match of following can give us easy marks. Regarding the mitochondrial cristae, identify the correct statements. Okay, so now the question will be related to the cristae, and they have been asked to select the correct statements. Okay, cristae, they increase the surface area for oxidative phosphorylation. Okay, increasing the surface area for oxidative phosphorylation. They are the enfoldings of the outer membrane. They are the enfoldings of the outer membrane. They contain enzymes for the electron transport chain. Okay, they are contain enzymes for the electron transport chain. This number correlates with the metabolic activity. Okay, so they are saying their numbers. So just tell me. So here they are the enfoldings of the outer membrane. So outer membrane doesn't have any enfolding, so we can eliminate the second option easily. Right? So in the remaining two options, we have seen that 1 and 3 are automatically it is right. So we have to study about this fourth statement. Their number correlates with the metabolic activity. We have studied this statement, and this statement is a right statement. Okay. See here, this first statement, they increase the surface area. Of course, it is increasing the surface area. And they are the infoldings of their inner membrane, and they are containing the enzymes for the electron transport chain, of course, and their numbers correlates with the metabolic activity. Of course, it is a right statement, right? Now, the fourth question is assertion succinate dehydrogenase is the only Krebs cycle enzyme not found in the mitochondrial matrix. Okay, see here, only Krebs cycle enzyme not found in the mitochondrial matrix. It is an integral component of the inner mitochondrial membrane. So what do we have studied? The enzymes required for the Krebs cycle will be present in the mitochondrial matrix, but there is an exemption, and that exemption will be the succinate dehydrogenase. It is present in the mitochondrial matrix. Okay, so the answer will be both A and R are true, and it is a correct explanation of A. Now, which of the following are true about F1 particles? That is, F1 particles means oxysomes. Okay, see here, they are present in the outer mitochondrial membrane. Okay, so they are having a base, stalk and head. ATP synthase is present in the head region. They participate in an oxidative phosphorylation. Okay, so we can easily eliminate the one option, because F1 particles is present in cristae, but here they have given the outer mitochondrial membrane. So we can eliminate the option B and D. Okay, so the second and fourth, these statements are right statement, and this F1 particles is involving in ATP synthesis, and that is nothing but an oxidative phosphorylation. So the second, third, fourth, all these were the right statement. Now, again, match the following question regarding with the scientist and the and their contributions with respect to the mitochondria. Kolliker, Altman and Benda. Kolliker, he is the one first observe the mitochondria in which type of cells? Muscle cells. Altman is the one named the mitochondria as bioplast. And Benda is the one named as mitochondria in place of bioplast. So the answer will be 2, 3, 1. Okay. So option A is the right answer. Now the seventh question, select the correct statement regarding the mitochondrial genetic system. Mitochondrial genetic system, so you should know mitochondria is having a DNA and it is circular DNA, and it is a right statement. Mitochondrial ribosomes are 80S type, so it is not an 80S type, it is 70S type. So we can easily eliminate the option 2 from the options. See, if we eliminate the option 2, just now the B option is the pending one. So we can easily, even though we didn't read about the option 3 and 4, we can easily come up into a right answer. Okay, so that is why option elimination technique will save your time as well as to save your to eliminate the confusion. Okay, so it is a circular DNA. Mitochondrial DNA is circular, and they are having an 70S type. Okay, 70S type. They replicate by binary fission is a right statement, and they are following the strand displacement model of replication. So this is a right statement. Now, so the another assertion reason question, mitochondria show maternal inheritance. Paternal mitochondria are selectively destroyed after fertilization. Of course, it is a right statement, but okay, so it is a right statement. Right? Now, which of the following which statements justify mitochondria as semi-autonomous organelle? So which justifies a semi-autonomous organelle? As they are having a DNA, of course, it is a right statement. 70S ribosomes is responsible for protein synthesis. Ability to synthesize all proteins independently. It is a right statement. It is not a right statement. Why? Because ability to synthesize all proteins. If mitochondria has the ability to synthesize all proteins, we can call mitochondria as autonomous organelle, but not the semi-autonomous organelle. But we are calling this mitochondria as semi-autonomous organelle. Why? Because they have the ability to synthesize some proteins independently. So we can easily eliminate the third option. Okay, so replicate independent or replication independent of nucleus. So whether this replication will be done with the help of nucleus or without the help of nucleus, as it is having the DNA, they are having the ability to replicate themselves. Okay, so this is a right state. So the answer will be option B. Okay. So the last question will be a correct statement about the mitochondrial DNA. So the mitochondrial DNA used to trace human evolution. Of course, it is a right statement. Mutates faster than the nuclear DNA is also right statement. Inherited from both the parents, and it is a wrong statement because it is usually inherited from the mother. Maternal inheritance. So we can eliminate the third option. Usually it is studying the recent evolutionary trend. So this is also a right statement. So the answer will be option B. So that's all about the question. So in today's class we have completely analysis the even very important instructions and important things with respect to the mitochondria. Thank you so much. We'll meet you in the next class with another organelle. Okay. Thank you.